It is increasingly plausible that specialized gut sensors, particularly neuropod cells, could act as a kind of internal early warning system, detecting biological threats like infection, inflammation, or even cancer well before conventional symptoms emerge. The gut is already the body's most densely innervated and immunologically active organ outside the brain, and it's constantly monitoring not just nutrients, but microbial cues, immune status, and metabolic byproducts. Neuropods, at the intersection of the nervous, immune, and microbial worlds, may be ideally suited to perceive molecular disturbances that signal early disease. Neuropods are a type of enteroendocrine cell, but with neuron-like behavior. They form synaptic connections to sensory neurons, especially vagal afferents, and can transmit information in milliseconds from the gut lining to the brainstem. While much of the attention to neuropods has focused on their role in nutrient sensing, such as detecting sugars or amino acids, research now suggests they also respond to bacterial metabolites, inflammatory signals, and immune activation. This positions them as not only nutritional sentinels, but potential detectors of systemic imbalance. The gut is the body's first point of contact with many pathogens, and the intestinal immune system is constantly surveying the microbiota for unusual activity. When pathogens or abnormal microbial metabolites are present, local immune cells release cytokines, chemokines, and other signaling molecules. There's emerging evidence that neuropod cells express pattern recognition receptors, PRRs, such as toll-like receptors, TLRs, including TLR5, which recognizes bacterial flagellin. If neuropods can directly sense microbial virulence factors or inflammatory cues, they may rapidly transmit a distress signal to the brain well before the immune system has mounted a full response. This could have practical implications for detecting gut infections, like Clostridioides difficile, in their earliest stages, before fever, pain, or diarrhea appear. Even more intriguingly, if neuropods are sensitive to metabolic byproducts of dysplastic or cancerous cells, such as abnormal lactic acid profiles or oncogenic bile acid metabolites, they could help detect colorectal cancer in a pre-symptomatic phase. Tumor microenvironments often produce distinct biochemical fingerprints, and if neuropods can respond to these via ion channel modulation or neurotransmitter release, they might act as molecular smoke detectors for malignant transformation. The communication route for these warnings would likely be the vagus nerve, which is increasingly recognized as a key relay not only for hunger and emotion but also for inflammatory states. In fact, vagal tone can be altered within minutes of gut perturbation, suggesting a level of reactivity that's fast and system-wide. This may help explain why some people report mood changes, fatigue, or altered appetite during the very early stages of illness, feelings often dismissed as vague or psychological, but potentially rooted in neurobiotic sensing. Beyond infection or cancer, neuropods may help monitor low-grade inflammation, such as in the early stages of inflammatory bowel disease, IBD, or metabolic syndrome. Subclinical inflammation in the gut can precede visible symptoms by months or years, yet may subtly shift how the brain perceives food, energy, or emotional state. Neuropods could pick up these shifts and relay them in ways that change behavior, perhaps leading to altered food preferences, sleep disruption, or anxiety-like symptoms. In this view, behavioral changes might be the first outward signs of gut dysfunction, not the result of it. This also aligns with the idea of the gut-brain axis as a predictive system. Rather than reacting to damage, it anticipates threats by rapidly synthesizing signals from diet, microbiota, and local tissue status. Neuropods could be central to this prediction engine, providing a moment-to-moment readout of the internal environment. If so, decoding their signals might someday allow clinicians to intervene early before disease has fully manifested. Technological advances may soon make this hypothesis testable. With tools like optogenetics, calcium imaging, and single-cell RNA sequencing, researchers can now observe neuropod activity in real time and identify which receptors they express under different conditions. If certain molecular signatures reliably precede infection, inflammation, or malignancy, it may be possible to use neuropod activity patterns as biomarkers, perhaps even captured indirectly via vagal tone analysis or gut-targeted wearable devices. In sum, the idea that neuropods could detect disease before symptoms appear is no longer science fiction. These cells sit at a biological crossroads, sensing microbes, sampling metabolites, receiving immune cues, and speaking directly to the brain. Their ability to respond quickly and precisely suggests they could be tuned to the earliest whispers of trouble in the gut. Understanding and amplifying this internal alarm system might offer not only faster diagnostics, but also novel preventive strategies for a wide range of chronic diseases, ushering in a new era of gut-based precision medicine.